All right, and then so just to reiterate, during sexual reproduction, the number of chromosomes matters. Diploid cells, all that means is that they have two full sets of chromosomes. They have chromosome number 2N, which is 2 times the amount our gametes have, which would be 23 in humans. N would equal 23, so 2 times N equals 46. And that just means we have a set from mom and a set from dad. Examples are human somatic cells. Then we have haploid cells. So you can think of haploid half. That's how I would remember it. And that just means those cells only have one full set of chromosomes, or chromosome number N. And they have only one set that is a combination of chromosomes from mom and dad. And I might have just lost you with that last bit. Yes, haploid cells have one set that is a combination of chromosomes from mom and dad. Don't worry if you're confused, it'll all make sense soon, I promise. And examples of these types of cells are our gametes, egg and sperm cells, which have N equals 23.
Please remember the differences, keep mitosis makes somatic sense. The cells that make up all of me, separate my sex organs, the haploid cells take half of me. Yeah!
If you ever forget which one is which, just think of those words in that song and it'll come to you. Okay, so this is just a chart kind of showing, it's charting through all the different chromosome numbers throughout meiosis. And so we start with chromosome number 2N. So we start with 46, 2 copies of each chromosome. And then also we've duplicated them. So we have, remember those sister chromatids over here, the 2 over here, and then we have the other 2 sister chromatids, and then those 2 and 2, they're homologous chromosomes because they all have the same genes on the same locations. If you cannot say that fast like I just did, then you do not truly know it yet. That is a great, I'm actually surprised I didn't mess that up. So that's a really great way to practice. Okay, so we started out with chromosome number 2N, 46, and then we, during meiosis 1, we split off into 2 cells, and then at that point they have chromosome number N, but remember they're still duplicated at that point, so there's still going to be those sister chromatids. Then those 2 cells then split off again to make 4, and at that point the sister chromatids will then separate, and we'll be left with just one single pair. No, they will not be duplicated at that point because they have been separated. So sexual reproduction, just being really clear about what that means. That just means, so organisms that reproduce sexually, like humans, like us, we fuse the genetic information, or the gametes, from 2 parents to produce offspring that are a genetic mixture of both parents. Not all organisms do it that way, but that is why we are so great. And so what happens when this occurs is a process called fertilization, which we've gone over. It's the actual fusion of egg and sperm to form a zygote. And then we remember then mitosis begins. That zygote then undergoes mitosis. It splits into 2, and then those 2 cells split off into 4, and then those 4 cells split off into 8, and then so on and so forth until you have a fully grown baby. So the purpose of meiosis, the whole entire purpose, is to make gametes, which again are sex cells that only have half the normal number of chromosomes. And we call these cells haploid because they only have one of each chromosome. These cells are used only for reproduction, sperm in males or eggs in females. There's no other purpose. It's the only reason. And so, during meiosis, each parent only passes on half of their chromosomes, 23, one from each pair, so that the offspring ends up with a full set. So you have to donate, mom donates 23, dad donates 23, and then the offspring will then end up with a full set, 46. So, the 2 different phases of meiosis, meiosis 1 and 2. Meiosis 1 begins with a separation of homologous chromosomes, and there is a reduction from diploid duplicated chromosomes to haploid duplicated chromosomes. I'm going to kind of speed through this, jam through this. Please feel free to rewatch, pause, digest at any point in time. Meiosis 2 is a separation of sister chromatids, and there's a change from duplicated haploid daughter cells from meiosis 1, and then those divide into individual chromosomes. So, before meiosis 1, is interphase. So a cell spends most of its life in interphase, which is the growth phase of the cell. And remember that it is divided into 3 phases. G1 or gap1 phase, the cell grows, makes proteins. And especially it makes those proteins that it makes proteins like DNA polymerase, because what's about to happen next? So S phase or synthesis phase begins and the chromosomes replicate. What do we know about replication? DNA replication. We unzip the DNA, helicase unzips the DNA, DNA polymerase then starts adding complementary nucleotides on one side, starts adding complementary nucleotides on the other side, and then we end up with 2 fully complete strands of DNA. We got an old strand and a new half, and we got the old strand and the new half, and then we have 2 strands of DNA that are both half old, half new. It's called semi-conservative replication. And so then the third phase of interphase is G2, gap2 phase. The cell continues to grow and make more proteins. And so at the end of interphase, the cell has 2 full sets of duplicated chromosomes.